Like, would it still work to create it?
Oh yeah, you could create it again with a new name.
Okay.
Yeah, it'll work. It's just now they'll have two DB map environments just called different things.
Okay.
And at a certain point, that's just a them problem
Mhm.
because they're trying to run your stuff, and if they don't know what they're doing, then I don't take a class. You must be this tall to ride the ride. But we try to do what we can to make it messed up. So I'm gonna get into Bora now. Bring back up. So our assignment 8 that we were working on, or lesson 8 that we're working through. Okay. So where we got last time was a rundown of like really basic metagenome pipeline, and then it got ugly really fast with all the choices. And kind of became paralyzed there at the last 5 minutes of class. We realized we have like 35 million different pathways through this metagenomic workflows. What I was trying to convey was that yes, there are that many, but nobody tries them all. It kind of depends on our question. And so what question are you trying to answer with your metagenomic data? That's going to influence things. And then if it's reproducible and you can justify it, then great, you're going to get through peer review and get that thing published. And if somebody has a problem with one of your little tweaks that you made, well, they're welcome to go change it in your code and run it themselves. And probably it's not going to change the answer. So let's take a look and let's design a workflow. And as we do it, we'll kind of see how to automate that on the install nonsense. So we've got one sample. We're just sticking with one for now. Keep it nice. It is raw shotgun metagenomic reads. So we are at this stage right here. Somebody's done the sequencing and we've got reads forward and reverse. We can assemble, we can annotate, we can bin, all that stuff that we talked about last time. And I just threw up some potential questions, but this is human gut metagenomes. And presuming that we would kind of run this workflow on a bunch more samples, what questions do you want to ask? Let me bring up the study page here. And this is the name of the study. Obesity, not cardiometabolic health status, drives altered gut microbiome functions in the metabolic unhealthy. Right, so they've got some people that are obese, some people with cardiometabolic health status problems, and they've got gut microbiomes. But that's why they were doing this sequencing, and that's the question that they set out to ask. Or it's the question that they realized that they could ask after they got their data back. What questions would you like to address? Because that's going to drive how we pick our parameters and everything. What do you want to know about the DNA that was in this tube?
I guess it doesn't matter more in the context of the question we're asking.
Yeah, I mean, that's what I'm wanting from all of you is like, what are you interested in? Do you want to know who was there? Do you want to know what genes were present? Do you want to try to find whole genomes in there? Is there a particular type of gene you're interested in finding?
I think I'd be more interested in community structure, so like amongst all these samples, what genes are consistently associated with the question, I guess, for this one was like obesity.
Okay, so what genes or what taxa? Because different taxa can have the same gene.
I guess we have more interested in taxa.
Okay, so you want to know who's there and like the structure of the community. Okay. No.
I think, just like the relative ones of like all the different species and trying to understand what's
Yeah.
actually in your sample.
Yeah, we can get relative abundance of different species. And that's like a super common thing for human microbiome work. We see people have diseases that are strongly associated with like a certain bacterial taxa kind of taking over.
So for relative abundance, is that you're using like read count?
Yeah, well, it's a bit different.
Yeah.
So if we do the meta amplicon, we can do read count. How many times was this read found? Here we've got a bunch of stuff, a lot of which is not informative to say who is there. And so it's the relative, it's the read count of the reads that were informative. And that's that coverage thing that we were kind of talking about, like where we've got it's sort of displayed here on the screen, the little short reads. So let's say we've got 3 different species present. For real, we don't know that yet, but we've got the contigs, those long stretches that we've assembled. Somewhere in that contig might be a gene that we care about. So whatever that gene is, maybe it's associated with disease, or maybe it's nitrogen fixation. You're looking at like wetland work or whatever. But also it might not be a real gene. It might be ribosomal RNA encoding spot. So for bacteria, 16S, for eukaryotes, 18S, or the ITS, the internal transcript spacer. These are kind of those like barcode regions of a genome that are really conserved because they code for ribosomes. What happens in a ribosome or on it? What's a ribosome do?
Protein.
Makes proteins. Okay, so without a ribosome, you die. You're dead. You never make it past the first cell division. So if you get a mutation in that ribosome, odds are done. It's nearly impossible to get any mutation inside of a ribosome that doesn't lead to instant fatality. And so that's kind of a perfect barcode that is well conserved. You and a fruit fly and a yeast and a mouse all have the exact same like 18S sequence, just a few little differences. And those differences we can use to like assign taxonomy. Well, it might not be a gene that we find there. It might be the thing that's encoding for a ribosome. It's not exactly a gene. If it's not making a protein, it's making a piece of RNA that folds up and acts like a protein. So if we can find that in there, then we identify that region and we say, okay, this is for the 18S encoding ribosomal RNA pseudo gene thing. How many reads map to that? That's the coverage. And that would give us kind of an estimate of how much of that species was around, or at least how many of its ribosomal encoding strands of DNA were present. This gets a bit nightmarish because how many copies of your internal transcribed spacer do you have in your genome?
Like too many.
Too many. How many do fungi have? Depends on the group. Some have 1, some have 300. And so they can look overestimated. If you get 300 copies, it wouldn't take as much of your DNA to look like there's 300 times of you. So this is why we call it estimating coverage.
Okay, let's drop another question. Can you use this to figure out what genes are being expressed? Like-
No.
Okay,
That's metatranscriptome.
Okay.
This is metagenome data. If we had metatranscriptome, they made cDNA, they extracted RNA instead of DNA, and then did a complement, and so they're sequencing RNA instead of DNA.
Okay, I feel like I'm just very confused about like this
Yes.
next step, so like. This is
Which
like,
one? Or the whole?
like most of it, but
Yeah,
like,
okay.
so we're starting with like the.
This is where we pick up.
This is where we are, right?
Yeah.
So we have that, and that's just like what they got from the machine.
That's just the files
Okay.
we're gonna download. They got it from the machine. They put it on the sequence read archive and now we're stealing it.
And then we have, it's like just like ATC, and we just like have that,
Yes.
and then. What does like assembly into, like, what is the big line of what are the tiny, like, I'm just
Oh, okay.
confused about like what.
So do you remember like in our FASTQ files, like it's just millions of short little reads? You've looked inside a FASTQ and it's like read quality score, right? Those reads aren't a million long, they're like 150. They only wrap like 1 or 2 lines. That's these short little bars here.
So
That's the raw
The big
reads.
bar is like the entire
The-
file.
The big bar is we call it a config. And it's where we've taken all the short reads and we've assembled them back together into longer pieces. So the long line here is the contiguous stretch that we assembled.
Okay, so like the first bit is all the little short reads.
Yeah, so
Okay.
our file there is just the short reads. When we assemble it, we get just long reads out. So we'll have like a FASTA file that's just
Yeah.
longer that we were able. Doesn't mean that that's what the real DNA looks like. We're trying to like get back to this squiggle spaghetti strands up here. That's the like full genes we extracted. We're never gonna get there with these. We can assemble them best we can and we'll get stretches.
How does it know, like, where does like which reads to put together or like,
That's that graph theory.
okay,
So the Kamers thing. So we get all the little, you don't need to know that. That's,
so like basically.
you're about to graduate.
I'm.
And I'm developing a class called genomic data science where we actually use that. That's graph assembly theory. You don't have to worry about it. A new generation of poor saps will have to.
So for my basic understanding, it should be like FASTQ has like all the little things, assembly things.
Puts
match
them
things-
together
match things
into
together.
longer stretches. So we're trying to like take our tiny little short reads that are at most 300 bases long. And we're trying to assemble them computationally back into this back into whole genomes. Because that's where we started. We actually had whole genomes in our sample. We shook them up, broke them up, sequenced. Now we've got little pieces that's just ATCG. We're trying to put them back together.
So is the reason for breaking it up to like determine if there's like other species in that sample set, or
The
like,
reason is a limitation of our technology. Our Illumina sequencing can only read so many in a row before it poops out.
I guess I'm just confused how we're going from like, if we have a full genome to going to a genome, like, I guess I don't really understand, like, if we are starting with like. If we say we're starting with the genome and then.
Yeah,
breaking
so,
it up and then
okay.
ending back at the same spot.
We've got a full genome. And let's say that it's eukaryotic, it's not circular, so we've just got a chromosome. Okay. And we would love to know the entire sequence of that chromosome from telomere to telomere. That would be awesome. But our machine can't give us that. It can only give us little snippets of 150 nucleotides at a time. And that chromosome might be 30 million. Oops, what do? All we can do is sequence as much as we can from across that thing. And then we're just gonna have a file that has a bunch of short little 150 long reads. The assembly step, we're gonna use a program called Spades. Tries to turn those short little reads back into the long chromosome. If it works perfectly, then we'll just get one contig. And it'll be, ta-da, here's your chromosome.
So it's kind of getting the details of that chromosome, when like you don't know the details initially. Okay,
Yeah, it sucks.
yeah,
If you already have a genome, you can match your short little reads right up to it with BBMap.
Mhm.
That's great. But we only have that like really high quality assembled genomes for a few species. So if you care about human genetic disorders, you're in luck. We've got a human reference genome. You get all your short little crappy reads from the Illumina machine and use BBMap, and boom, here they are. This one matches up perfectly, except for this one base pair difference. And guess what? We've also found all these reads that match up perfectly to that same spot, and they all have that difference too. Well, what are you talking about then? Your sample is different in that one nucleotide. And that's a SNP, single nucleotide polymorphism. That's how we find all kinds of like genetic disorders, mutations that are linked with diseases. If you grab a sample of soil and extract all the DNA from it, you're in a much different world now. What reference genome are you going to use? You have 1,000 species of bacteria in that pinch of soil. So you would need 1,000 reference genomes to match it against. And half of those are probably due to science, undescribed. Some of them have been seen before, but only by their DNA. Nobody's like seen it in a microscope, seen what it does, given it a name. So that's where we find 99.999% of all the work that we're going to be doing. It's not going to be, I'm looking for a single nucleotide polymorphism associated with this type 4 glioma tumor. You work in a cancer research lab, yeah, that might be your life, but boring. So boring. What's even the point of you? Like, honestly, that job's gonna be gone because it's so easy at this point. Here's the reference genome. The data goes straight from the sequencer through a pipeline, matches it up, highlights the SNPs. Done. It's seriously not a career for that now. Imagine 10 years from now. So really, yeah, it's done. And so it's just a metagenome. We have that problem of there's multiple species in our sample. And now we're going to try to assemble all those chromosomes at the same time from many different species. So we can't do it by like matching them up against a reference. We have to do it de novo. If we have really long reads from Oxford Nanopore, then we can do that overlap where we can say, oh, these 1,000 bases like match up perfectly together. I bet they go together. And the best genomes now that we're getting are from Oxford Nanopore, but they don't get enough reads to do metagenomic work very well. For a reasonable amount of money, put it that way. It's still a bit expensive. We can get a human genome for about $200 now, fully sequenced. A metagenome from a single pinch of soil would be, I don't know, I'd probably budget 6,000 or 7,000 if I was doing Oxford Nanopore for one sample.
Oxford
Nanopore, Illumina is what we do for human genomes now. Because it's so we have a reference, so we don't have to assemble and go through all that. It's just put on.
But you need so much with Oxford Nanopore, you need to so many to get enough.
You need so many, you have to sequence it so deeply to get enough reads to cover the whole genome.
what's the difference between the two? I know we've gone over this.
A little bit. We haven't seen Nanopore data yet. It's just a lot longer.
Like the little green bits, like they're more than 300 bases.
Oh yeah, they are indefinitely long. And the technology is really cool. And it's getting, it suffered for a few years from being like error prone. See if I can find an image of the little process. It's a pore. It's a nanoscale pore. Developed in Oxford, England. So hence the name Oxford Nanopore. But what they've got is like these magical little pores that can suck up a single strand of DNA. And as it gets sucked through, it reads it. So it uses like the electrical properties of A, T, C, and G, those base sugars, as it reads through, and it measures that electrical current, and then converts that to A, C, and G.
It's gonna be a dumb question, but is it? From the picture, it kind of looks like it runs like a gel, or is that not how that works?
Yeah, no, it's not a gel.
No, yeah, but
Yeah.
like the premise, I guess, is one that.
Yeah, sort of. Yeah. So it's pulling the DNA through, and as it gets pulled through, it's going to be just like reading it as it goes along. It's cool technology. It's just limited by the number of pores you can put on that chip. I'm not sure what they're doing there. They're probably talking about epigenetics for this particular image, because they can read epigenetic stuff too, which is awesome. Because Illumina can't do that. Yeah, so it can detect like the methylation profile, see what's being expressed, what's being hidden. But it really is pulling through a single strand of DNA and reading it as it goes through. And that means that it's not limited to short little stretches. So the longer strand of DNA, it can pull it through. There's even global competitions for DNA extraction now to see who can get the longest DNA reads. And we're getting up past 1 million bases. Yeah. And those are already, that's better content than you would get from assembling things.
So Illumina just needs to be really tiny because they just.
The technology itself, the way that it works, we won't go into the details, but it limits it to like 300 bases at most in a row. Yeah. And then it actually just kind of gets like worn out. Gets tired, you have to start over. That's also why the reverse reads are always worse, because it's like tired by the time it gets to the reverse part. Tired is not the right word, but we'll stick with it. And here's the Oxford Nanopore machine. They've got an iPhone version now too that you can use in the field.
It's crazy.
No amplification.
Just like accurate on the side.
Isn't it cool? I bought one of these, it's $900 for this little thing, which is very cheap for a DNA sequencer. But then they're kind of a terrible company to work with. Everything's proprietary, they're worse than Apple. And you have to buy their kits, and it wound up being like super annoying expensive. Things expire in a month. So now we've got these other versions of it. So we've got the iPhone version, the laptop version, and then the Prometheon and GridION, which is just a bunch of those. This is what happens if you send it out to a sequencing facility and say, I want nanopore sequencing, they'll throw it on one of those. And it's still 1% of the amount of data that you get from a film. So if you want to do a really good assembly, you've got like one species, like you got a fruit fly, you clean it up, get all the bacteria off, smash it up, extract DNA. You would do a hybrid where you do Oxford Nanopore a little bit to get those long pieces, and then you use Illumina to map it to those and build it out. In practice, it's probably going to be 20 years before this becomes cheap enough to do the amount of work that we put onto Illumina. And so the bulk of metagenomic and metatranscriptomic work is going to be that short Illumina reads that we have to assemble. Okay. So those long bars are the contigs, the short ones are the raw reads, showing how many times each one maps to that config. And that gives us an estimation of coverage for that particular locus. If we want to do a taxonomic profile, we technically don't even have to assemble and build contigs. We've got our short little reads. We can take those short little reads and map them as they are against the database. We can do that and skip the whole rest of this process. Great. So Rosie, you want to know the community composition here?
Yes.
That's the easiest question to answer.
Right.
Yeah.
Yeah.
So basically, yeah, we can kind of like blast those against the database and see who's there in what abundance. We got a problem though. Of course, that most of those reads are going to be not phylogenetically informative. So they'll be useless.
Is that because they're so short?
Because they're so short, yeah. Or they're from a region of the genome that's too conserved, completely identical across all species. Or it's from a region of the genome that's like a telomere. Cool. ATTA, TTA, ATTA, TTA. Same for you and a fruit fly and a pine tree. Can't tell them apart. Okay. So what do we do? We wind up throwing out the bulk of our data. That's okay. The other problem, you thought there was just one. There's another problem in that in order to map it against the database, we have to have the things in the database already. That means they had to have been studied by somebody, some poor sap in a lab. Their hands getting pruned from wearing gloves all day for months, culturing this bacterium, giving it a name, sequencing its genome, adding it to a database one at a time. People have done that, but they've only done it for some bacteria, some fungi.
I have a question, but like, I guess on a time scale, how long does it take to sequence the genome of a newly described species? Like if I was a student and I said that's my goal was to find these species and actually put a name and sequence the genome, how long did that take on average? And that just like widely ranges.
I mailed it off to a company called Seedcoast, and 10 days later, I've got a genome.
Oh.
But that's one at a time.
Mhm.
And the culturing work's what takes the longest. So you're relying on somebody in the past having done that work for that particular species and adding it to the database. Cool. This is why I chose the human gut metagenome for this project, because that's largely been done for all human gut stuff. Get out of that system, it's over. Game over. It's not in the database. So we'll do a taxonomic profile and we'll get back 90% undescribed bacteria. Useless. But we can do it. And I bet we get a good answer here because it's human gut. There's only so many species that are associated with humans. They've been well studied because it's biomed. There's money to do that. There's hordes of undergrads willing to work for free all across the world, culturing bacteria from poop samples. glamorous work. I've done it once. Done poop samples and I've done like dental plaque. Never again, it's too much paperwork. Also, it's really boring. Stinky. All right, so we can do that. That can be one of our things. So we can do this. Who is present and what is the community structure? Evolve that, folks. Any other questions?
What
You're
does,
not limited to these, so if there's something you're curious about for human guts.
what does in fixation mean?
Nitrogen fixation, so there's only some species on Earth that can take nitrogen out of the air and turn it into a usable form. So fixation is that process of turning a mineral form into organic. So carbon fixation, plants do that. They take CO2, turn it into sugar. That's fixation. Bacteria can do nitrogen, some bacteria can do nitrogen fixation, where they take N2 out of the air and turn it into what's the usable form? Oh. Yeah. Well, yeah. NH3, NH4. Those are our usable forms of organic nitrogen. That's what plants can take up. So if the bacteria weren't doing that, there wouldn't be plant life. It would all be dead. And so it's of great interest for us to identify in fixation genes. And they're really well characterized because there's only a few ways that you can do that. And they tend to be from anoxic environments, so like below the sediment. It's happening on the ocean floor and stuff.
What is the lignin?
Lignin is what trees are made out of. So it's what's left over after the tree is dead. It's cellulose and lignin. And those are really complex carbohydrates. So a tree sucks CO2 out of the air, turns it into sugar, and then uses that sugar to be a tree, and make more tree and grow bigger. It's laying down cellulose and lignin to do that.
How does that connect to back to
You eat cellulose and lignin all the time. Every plant you've ever eaten is full of cellulose. And there's lignin in there as well. Can you digest that? No, there's no animal on Earth that can actually eat food. What about termites? No, it's fungi living in their guts that's doing that. Right? You work with ambrosia beetles, okay? So cows eat grass. They can't digest grass. It's fungi that are digesting the grass in their rumen. That's why they've got the multiple stomachs. They gotta like ferment it. So no animal can actually live on plants by eating cellulose. So that kind of function is of great interest for animal systems as well. Particularly in the ag industry, we're looking at like in the rumen of cows, like how does this change the lignin digesting genes in the cow diet, right? So if we put cows on a new diet, this is gross, but there are feedlots that feed their cows like old Halloween candy and stuff. I'm not kidding, like seriously, like buckets of gummy worms. It makes them fat really fast. It's just sugar. Sugar will make you fat really fast. Farmers know this, and so it's an easy, cheap way. If people put high fructose corn syrup in their cow feed, fatten them up. Yeah. Fun stuff, right? But guess what? There's also been research that's like coming out saying like, oh, you might not want to do that actually, because like that's really horrible for the cows. It's very painful, it fills them full of gas, they have like miserable existence. Yes, they get to eat gummy worms, which is awesome, but then they like are bloated and it destroys their gut microbiome and they can't go back to eating corn and grass.
Is that also increasing the things?
Uh-huh.
That's great.
Yeah. So yeah, so there's a fun question. We could talk about like methanogenesis genes. Those are well characterized, so we can look and see like, what's the potential for methane to be released from this sample versus this sample. Right.
And for all these questions, are we like comparing it based on the patient profile?
Yeah, so eventually we'll like be able to do this across 100 different patients and see if there's significant statistical differences between like obese versus non-obese patients, that kind of thing. Yeah,
Like that's what we should do.
that's the. So for assignment 8, that's the sort of thing that you're going to be doing. But we're going to try to define like, what there's so many things we can do with all these short little reads. We can profile the community. We can go in and look for specific genes. We could try to get the entire metabolic potential of that community.
And I feel like just like genes.
So we can pick like a few key genes. And this is also really common because if you're trying to get the entire metabolic potential of the community, that's really hard. There's a lot going on. You have to look at all those KEGG pathways and all that stuff. Well, you probably don't actually care about the ability of that individual to do one of those random little transformations of this substrate to that one. You care about that key step, whether it's the NIFH gene for nitrogen fixation, or if it's lignase. Lignase, yeah, I think it's lignase, whatever. Can this community digest a tree? We recently found that in a few bacteria, but it's pretty small and they don't seem to be expressing it. The only things that can digest lignin are fungi. So that's how termites work. That's how cow guts work.
So like for the relative abundance in genes, you're kind of looking at all the different all the different bacteria and like
All the bacteria.
they all, again, this is like maybe basic biology
Yeah, no,
right
you're
now,
good.
but like are there like specific genes like you can tell from the sequencing like this is for lignin and this is in like 80% of them, so they
That's
can
it.
do this. Okay.
Absolutely. So
Okay.
we can recognize if that gene has been studied, and a lot of them have, because of basic metabolism, and
Yeah.
we're all the same. We use the same stuff.
So
We just use slightly tweaked. It looks a bit different on paper when we like write out the amino acids for our glycolysis genes. They're almost identical to a yeast. And this is why we can study human disease using yeast without having to like, murder humans by the millions to like cut them up and stuff, right? We can do it with yeast and we can like put them in a salt bath and see how that changes how their genes are expressed. And so, yeah, we actually have good profiles of like a lot of genes. I wouldn't say most, because there's still so much unexplored biology out in the world. It's still the Wild West. If you go to a soil sample, good luck, right? There's just all kinds of crazy genes out there. There's a whole branch of biology called bioprospecting. And this is kind of like a biotech sort of thing where we go and we look for cool new proteins that can do financially useful things.
You know how they found like the PCR?
PCR, Taq polymerase. Guess what? We all have DNA polymerase. That's for copying DNA. But we don't live in a hot spring. Our Taq polymerase, or our polymerase, can't survive at high temperatures. We get cooked. But there are some crazy extremophile bacteria living in Yellowstone in their hot springs, like right near the boiling point. They also have to copy their DNA. If they don't, then they don't get to live there. They don't get to make babies, right? So natural selection has given them a slightly different version of Taq polymerase. More or less, it still looks like Taq polymerase, but it uses different amino acids that don't melt as easily. So they can survive at 92 degrees Celsius drop you in that, you're cooked, right? It's over. You're not gonna last a second, you're dead. But that means that their Taq polymerase can operate at that high temperature so they can survive PCR. And that won the Nobel Prize, that's bioprospecting. And it's revolutionized all of biology. Lactose-free milk,
What's the name?
lactase. That's an enzyme that most humans make, not all, but most do. And definitely almost all humans from birth. If you can't digest milk when you're a little infant, guess what babies eat? Milk. Okay, so we got lactase, but we want to add that to milk to get rid of the lactose for people who are lactose intolerant. Problem is, our lactase requires about 98 degrees Fahrenheit to work. You heat up milk to body temperature and let lactase work on it, it curdles, it tastes bad. So researchers went to Antarctic soils and they looked for lactase-like genes from the bacteria there that operate at very cold temperatures, and they found them. That's $4 billion a year industry now, is cold lactose removal from milk from bacteria. It's so cool because like we know what lactase looks like in a human. We kind of know like what the sequence looks like. Amino acids are going to be more or less in this order. When they fold up, it's going to be this shape. We go looking for that same thing in Antarctica, and we got ourselves a cold tolerant version. And so yeah, we call these like profile of a particular gene. Like we kind of know its signature there in the data. We can train a machine learning algorithm to detect that signature, even if it's slightly different. And that's what our annotation set is going to do. In the background, it's running some complex machine learning called hidden Markov models, where it's been trained on all these different genes and kind of like it learns how they look. This is that neural net type work where we're kind of learning what Tweety Bird picture looks like, and you give it a new picture, and it's like, this reminds me of Tweety Bird. Cool. So that's, once we've got our contigs and we find our genes, then our annotation step is going to go in and say, this looks like the NIFH gene, or it is exactly the NIFH gene from this species. So that's the annotation step. And so we can do that, and we can absolutely do it for everything, or we can say like, we're actually just going to be looking for this one type of gene, whether it's cellulase or lignase or whatever, lignase. So we can do that. There's not going to be much nitrogen fixation happening in the human gut. But maybe some, but we could do it for cellulase. Okay. So I think 2 questions is enough for us. That'll get us through this whole pipeline. So now let's think about the steps that we got to do. And we're starting here from our raw reads. We need to, what? Clean them. We got to, well, let's back up even further. We got to download them. No, we got to back up further. No passcode. We got to set up our environment. Then we got to download the reads. Then we got to clean them up, and only then assemble. And then we annotate. And then we can go like run our actual, then we can answer our question.
Would you clarify what annotating like in this example would entail, being that you said like so for the first question, like the like who is present, you said that's just assembling, not
Yeah, the who is present, we're actually just gonna use our cleaned up short reads, and we're gonna essentially kind of blast them against the database of human gut bacteria.
Okay, so it'll be able to like look at like the little 300 base
Mhm.
pair and be like, oh, it's this one,
Yeah.
it's this order. Okay,
Yeah, it'll try its best to get to species, but if it can, it'll give us like a genome or like a genus. Be like, yeah, I couldn't tell the species, but it's definitely this genus.
and then we need the assembly annotation to get to the gene finding, so the assembly is the like make a longer thing.
Assembly makes longer stretches because there's no way the gene for cellulase is 150 nucleotides. It's going to be thousands of nucleotides long, so we got to get that long stretch.
And then the annotating is,
And then the annotating is once we got the long stretches, well, now we got to find start and stop codons on there and actually find the gene. And then we get to take that gene and match it up against the database. Only this time we're using like not just perfect match, we're looking for that signature. It looks like this gene. It's in this gene family.
so the annotation is gonna get you like all the different genes in your sample, and then after that, it'll be like, I'm on cellulase, and then look at that.
And it's really nice when we get to pick a few genes that we care about rather than everything. Because yeah, it's a lot of data. This is just one sample. We're doing a study, we've got 100 samples. That's 100 annotation files. If we could just like go through and say, just give me the ones that are in this gene family. Perfect. Now we've got small files and we can put those into our Python and just actually do our stats. I mean, at that point, we're like done with the bioinformatics stuff and we're moving into biostats. All that's left on this pipeline here are the steps we won't be doing, where we try to like bin those contigs by species and then assemble them again into full genomes. We could do that. We could try to reconstruct entire bacterial genomes out of our mess. But what's the question we're answering for that? Neither of our questions require that. Like, I don't care if this gene came from a whole genome or not, like the gene was there. That's what we're interested in, so we can kind of stop.
What's like a use case of like someone actually like trying to assemble the full genome? Like would that just be like hyper specific study, you know?
Yeah, so the vast majority of bacteria aren't culturable yet, like meaning nobody has figured out how to grow them. And so we can't grow them, so we can't grow them up and just get them and get their genome. So the only way to get their genome is to assemble it out of the metagenomic data.
And then you're just kind of trusting that you're assembling at that point. And you're
Well, we can look at our assembly and see how complete it is. See if it's been circularized, all that kind of stuff. And so the majority of MAGs, metagenome assembled genomes, there's tens of thousands of them available, but they're like 70% complete, 80% complete. That's publishable. It's like a draft genome. So that, let me see if I can find the use case. I think get the actual name of the paper. This one will do. Yeah, a superbug. So antibiotic resistant. All right, so this approach neglects sources that are not amenable to microbial culture. So boom, we're studying like antibiotic resistant bacteria that we haven't figured out how to grow. Don't need to if we've got metagenomics. We can get their genome anyway. Yes.
So earlier you said, like, if you send to. Yeah, exactly, but you send to a facility, you said like somebody's like, you're bringing genome back. Are they doing the vending, like, or what is that in the
Yeah.
context of this?
They're just gonna. Oh, for like a full genome?
Yeah.
No, they're just gonna give you your raw data. But then 2 days later. The genome. Keep in mind that I'm not writing pipelines from scratch every time. Once I get it to work on one genome, I'm done. I get new data. I point my pipeline to that file, walk away. I mean, I could have given you my entire pipeline. Just cancel class for the rest of the semester, but here, do this. But that's not gonna work in every test case, right? That works for my little yeasts that I like to culture. So here they've discovered an entire new phylum of bacteria. A whole phylum. That's like suddenly discovering that animals exist. Like all we knew about was trees. Like, guess what? Scientists discovered that animals are here too. Everyone's like, what the hell? Yeah, there's so much novel diversity that's still undescribed for bacteria and fungi, like the microbes, that we're discovering entire new phyla by doing metagenomics. That blows my mind. I think it's awesome. So that's going to be the two questions I think we should address. These are nice tractable questions. Who's there? And then we'll look for cellulose genes. Cellulase genes. Sorry. Yeah. The software we're going to have, I've got listed here. You've already got SRA Toolkit. You've already got BB Duke. You don't know it because it's called BB Math. Okay. That's another one of those. Spades, Proca, and Metaplan 4. I was hoping that nobody would want to do metabolic reconstruction. So I didn't even put that software here. It's great. You guys chose right. Okay. So we're going to have the scripts shown here: setup, download, quality control, with Duke, assemble, annotate, then we'll get coverage. So I guess that is SAM tools. We've got that one already. Coverage. And then I think what I'll do is give you my first little script, see if you can get it to run your own. Okay, so I've got just my accessions. That's that one accession from the lesson. Pipeline just runs these. Here's my setup script. It's not doing anything too fancy. Telling it where the scratch space is. I'm creating a project directory for this project in the scratch space, and a separate directory to store databases, because we're going to need databases to like match our stuff up against. Then I'm making the directories: data raw, data clean, output, and then in the database directory, one for Metaplan, one for Proca. So that's it. That one's nice and simple. I will Copy that. Put it here, and then reduce the size so it doesn't take up.
And then, so like,
Oh, that was my wrong script. So that's my setup, and then the downloading. Look at this one. Again, I'm saying where the scratch directory is, where things are gonna be. Saying like, here's my database directory, okay, great. There's a place to put my sequence from the sequence read archive, then scratch base SRA. Then I'm doing a little if. This is the test. It's testing if that directory exists. What's the double lines? Kind of like an or statement, but this is for a test case. It's saying if this fails, if it returns exit 0 other than 0, then run this. So that little line there, this means if this directory doesn't exist, then make it. Okay. All right, and then I'm running a for loop, even though I've only got one. I want this to be good to go for when I have 10 samples. If this file doesn't exist, then, and that file is my sequence read archive, the accession number, then the accession.sra, then prefetch. So I'm going to pull in that SRA file. I want it to go in my SRA directory. Here's the accession from my for loop. So if it doesn't exist, then download it and then extract it and actually get the FASTQ files out of the SRA file. And feed, done. Okay. Then I'm going to compress them, and then I'm going to install the Metaflan database and the Crooked database. I'm going to give you this as well. You can get that to run. Beautiful. Nice and readable. Okay. So between now and next time, see if you can get all your software installed for this and get your stuff downloaded. Next week, I will be in California for working group workshop-y thing. So no class,
on Tuesday, well, Tuesday's election day, but on Thursday.
both days. No, I'm gone all week, so no class next week. I will be putting videos up as usual, so I will like kind of go through this and then go through creating the next script, quality control.
So between now and like, not next Tuesday, but Tuesday after that, but
No, you can have this done by next Tuesday so that you're ready to follow along with the video.
so the videos are gonna
The
release.
video is going to start from assuming that you've got all these tools in your path and are ready to play. You've got your data downloaded. Yeah. I mean, I guess you don't have to. I won't be checking. But if you want to follow along with the video that I hope will come out like Monday or Wednesday or something, then
We're doing the first one. Honestly, never really talked about that.
You haven't?
No.
No.
Seriously?
Oh, I
use it
was all the time.
Yeah. Oh, you're killing me. Okay. In biocomputing, our main directory for the entire class, there's class recordings right here. Look at that. Yeah, YouTube links. Thanks. Like and subscribe. No, I just said like it.
Oh, we can't get your comments.
You can't get in my comments. I don't want that. You've read YouTube comments. I want nothing to do with that.
You don't monetize your videos?
No.
You don't-
I don't get enough views. Who
You
wants to
don't
watch?
want political ads?
Yeah,
Where are you going?
Santa Barbara. Could be 12 hours a day of data harmonization. How do they